You want to start recording? Excellent. So welcome everybody to our first Industrial Biotechnology Innovation Catalyst webinar. This is the first webinar in our new series where we'll be launching a new session every month. Every Thursday of every month. So please look out for the forthcoming programme. But just to introduce myself, I'm Aileen Miller. I'm Professor of Biomolecular Engineering within the University of Manchester, and I am Director of the IBIC, our Industrial Biotech Innovation Catalyst, which is a £5 million EPSRC funded impact acceleration project, which is all about trying to get the research across our Northwest institutions out into industry, whether in partnership through knowledge exchange or in the formation of spin-outs and startup companies across our region to really help grow our bioeconomy. So if you are here and are interested in tapping into some funding that we have available, please look out for that. Have a check out of our website. We've got information about the current open calls for research projects in collaboration with industry and or secondments of staff into or out of companies and across that industry academia interface. And we have got innovation awards that are going to come live between now and sort of the end of this calendar year. So please check out the website for the latest news and sign up to our newsletter. And without any further ado, I'm going to introduce our inaugural speaker for our webinar series. It's great that we've got two excellent speakers online today. The first is Ali Diaz de Rienzo. So Ali is going to kick things off in terms of talking from lab to life, and she's going to be followed by Daniel Wales, also from the University of Salford, who's then going to go in a deep dive about one of the sort of project areas. So really pleased to have you here, both of you today, Ali and Daniel. So Ali, I'll hand over to you.
Thank you, Aileen. Let me put the full show slide. All good?
Perfect.
Yeah, amazing. So I was going to say good morning, everyone, but it's already past midday, so more of a good afternoon. I am Ale. I work at the University of Salford as a lecturer in biomedicine, and I am part of the IBIC as one of the partners of this Industrial Biotechnology Innovation Catalyst. And today is the first webinar series out of many that are to come, as Aileen said, every Thursday. So keep an eye on the comms. I have the pleasure to share the first one with Danny, which is our PhD student, and is one of these people that is going to be translating what we do in the lab to life. So since the beginning of my days of research, I've been doing biotechnology. I'm a microbiologist by training, but more focused into industrial biotechnology. So my pathway in the UK has been as a postdoc, first sponsored by Unilever, then back in the day used to be a TSB between Unilever and Croda. Then I moved to the University of Manchester, where I did my second postdoc on process engineering at the chemical engineering department. So everything with the purpose to finding these molecules that we call antimicrobial molecules that could have a wider application, not just on the home care industry, but in the healthcare and other industries around. In 2022, I joined the University of Salford. The University of Salford main campus is located in Peel Park, where we have the School of Science, Engineering and Environment. It's a big school, and we are about 400 academics. One of the labs of one of the main centers of the SEE. This school is represented by different academics that do research from nanoparticle design, which is led by Dr. Shelka in the university. We have an amazing group as well that focus into phage bacteria interactions, and they love to translate what they do in the lab into real life settings, including a big group on public engagement, which is led by Professor Chloe James. So if you haven't had the chance, go to the Manchester Science Festival and go and look for the Drug Tech, where you're going to have an amazing time learning about phage bacteria interactions. You have another research group that is led by Professor Nirshini Nirmalan, and is their core research is on fast track antimalarial drug discovery. And it's a big group. They have a wider collaboration with the University of Manchester, with universities in Kenya, and with protein technologies here located in the UK. Another group is focused on environmental metagenomic, is led by Dr. Alice Risley, and focus in doing this relationship between ecology or microbiome ecology and evolution, and their effect into the environment. Another highlight is the slug microbiome group, which is led by Dr. Natalie Ferry, and they focus in the characterization of microbiome of herbivores. It's not as nice as it looks in there, because these tiny consortium are very particular, but this research is widely funded by and led to a good collaboration with the Erlang Institute and another biotech companies into the University of Salford. Now, there is not just research around environment and science, but we have specialist facilities at the university that comes with energy house labs, acoustic testing laboratory. We have the NERIC, which is the North of England Robotics Innovation Centre, and is a 60 million pounds robotics and AI facility that helps these small medium enterprises to gain a competitive edge through the application of this intelligent automatization and robotic solution. Here in the pictures, you can see our tiny dog, the yellow tiny robot. He's moving all day across campus, as an example of the amazing things that we can do at NERIC. We have the Salford Business School that comes with the Center for Sustainable Innovation, which I'll mention later today. But then where I am in the whole picture, right? So as I said before, it's a huge school, and we have different areas and research pathways. But this tiny cup here full with beer is what we call the brewery. And that's our big biotechnology these days achievement in terms of the school. All the amazing things that we've been doing, we want to do, and we have done already in terms of innovation, especially to develop or to keep enhancing the industrial biotechnology here in the Northwest. So, Salford, the University of Salford is the only university in the Northwest that has a brewery into the main campus. This brewery is a subsidiary of the university and was designed as a hybrid between a commercial and an academic enterprise. In another words, we use the brewery as a hub for knowledge exchange between our different programs and the production into a real pilot plant. We are working closely with the Old Fire Station. That's the full name, Lark Hill Brewery and the Old Fire Station Bakery. And we're working close with them and trying to exchange or facilitate these the skills that could be passed from the people that works at the brewery to our students either a level 6 biotechnology program or a level 7, which is our master's in biotechnology. Using the Hill as a commercial partner, and we took advantage of these our facilities, and we would like to provide opportunities not just for our students in terms of knowledge exchange, but for industry collaboration. And that has led us to use Hill for different projects inside of the university. We have different partners for advanced research into the brewery sector and more general food and beverage sector. And we have founded a hub for facilitating this knowledge exchange through the Microjoule Brew Lab, which aims to provide cutting-edge brewing technology, starting from the Northwest and then expanding through across the UK. This lab provides chemical analysis, biological analysis, sensory panels. We train our students in not just microbiology safety, but in the whole range of analytical skills that are needed for quality control within the food and beverage industry. We go out there and promote and do outreach and public engagement with our brewers at this moment into the Great Manchester area and expanding to a wider industry collaboration. As an example, last week we went down to London, part of the team, to the trade and expo drink that was hosted at the London Excel. So we're trying to expand our connections from the Northwest to the UK. If you want more information about what we do, follow the link, jbl.co. I'll give you, at the end of the presentation, a quick look how our website looks. And one of the projects that we are working at the moment in Microjoule Brew Lab is the BMAP. So we are collecting samples, or we already collect 24 samples across 24 different breweries in the Northwest, with the aim of providing a BMAP that will help us to determine the species of each yeast sample and model their evolutionary relationship and identifying the genes of interest and if they have an impact in the final products that all these breweries produce at the end. We aim to give back to them as a thank you for giving us your strain, this BMAP, so they know where in terms of evolution their strength is placed to produce the beers that they sell and make at their own breweries. So that's an ongoing project. We have not just the brewery as the final product, which is the beer, but we are involved in other areas of the brewery industry. In this case, we had a project with Beer Piper. Beer Piper is a company that provides cleaning solutions for the brewery industry to clean their pipes after use. So Beer Piper was committed and to look out to develop better technologies to improve the cleaning on sustainable technologies, which means reducing the chemical components of their formulation and provide a better approach in terms of sustainability. This project started as, this is our original formulation. Can you improve that formulation in terms of cream approaches? And everything was, and this is one of the typical examples of how things move from the lab to life. So everything was originally designed to study that effect on cleaning in a lab bench scale. So we will try to clean small tubings that has been formed with some sort of biofilms produced by Saccharomyces and see how the new product was using. However, one day, just brainstorming with the team, we say, why do we need to do this in the lab if we have pretty much a pilot plan across the road that produces beer, sells beer, and which pipes needs cleaning anyways? So we have the product, we have the pilot plan, we can translate the effect in real life settings. So one of our students, PhD students, and evaluate this effect of the new product in real pipelines after the dispense of beer. So this is our PhD student, Sean. Sean has used the new product and have cleaned about 8 lines, different lines, and analyzed the outcome in terms of the engineering of the product, the chemistry behind, the green approach, and the microbiology residual after the cleaning process. Everything was done in a large scale. So that was one of the main key points out of this project, that we could use Lark Hill and the old fire station as a real world test facility to examine the effectiveness of nanobubbles as line cleaners without disclosing in more detail the project. In this project, our colleague Shelka, which is the lead of the nanoparticles lab, was involved as much as myself and another colleague from the engineering school. Now, we are involved on the final product, which is the beer itself, in terms of quality control and microbiology testing. We've been involved in the cleaning of these pipes, but we know these days the brewery industry got so many challenges. And the main challenge is trying to approach the market and try to engage with this sector when the struggle is real, because even if it's the food and drink industry in the UK is the largest one, the microbrewery has been really affected after the pandemic. So in terms of sustainability, they would like to reduce the environmental footprint, but that has a cost associated. They would love to improve the quality of their products, again, cost associated. The waste management, because they would like to minimize the production waste. Why? Big companies, as in Heineken, Carlsberg, and among others, they process their own waste. Medium enterprises, they have agreements with the farm industry, so the agriculture sector will take that waste and use it for farming, which is a balance in terms of costing. But the small breweries, and an example is Lark Hill, that waste needs to be removed from the brewery every time that a production cycle is being done. And that has a cost associated, which then has an impact in the overall production of the beer for small breweries. That's one of the main industry challenges, as well as the employability resources. If the brewery is too small, then they just cannot afford to employ so many people, but through internships, capability of more students that are engaged and aligned with these type of sectors, we could create job opportunities for them in a more friendly way when you prepare the next generation to work in the brewery industry. In this sense, we realized that one of the biggest challenges was the waste, but it's not just for the brewery sector. So we are very, very focused into explore the possibility of producing high valuable chemicals through green routes. And we have, at the moment, a collaboration with a couple of companies that are interested into anaerobic digestion of the cellulose, hemicellulose residues from paper waste. So this is an ongoing project that have been facilitated by the Center for Sustainable Innovation, that is led by my colleague Silvia Tedesco. And we have a brilliant research associate, Osgun, working on the project at the moment, and it's an ongoing project because we are expanding our aim to a sustainability approach as well as into the food and drink sector. So this project is an ongoing project, and in other words, what we aim is taking the paper crumb and produce energy through that paper crumb. Now, that's not the only waste. One of the main issues, as I said before, for the brewery industry is waste, especially for the small microbrewers. So this, I'm not going to disclose anything else from how we hope to manage this waste in terms of the production of high chemicals, because that's what Danny is going to talk briefly after myself, but it's one of the aims of our hub as a group as well, trying to provide as much as we can in terms of chemicals, biochemicals, using different type of waste. So far, we got engaged with an industry that produces paper waste. Now is the brewery waste. And we have incoming projects, not just from the brewery industry and the paper industry, but we've been approached by the coffee industry because there are a lot of value into the spent coffee, especially in the spent, and that spent coffee got the properties to produce high valuable chemicals as well. So we are talking with a company that is based in Macclesfield, which is part of the Northwest, which are very interested in providing this spent coffee waste and see how could we work with them for the produce of more sustainable chemicals, which is at the moment very exciting for us. In terms of sustainability, most of projects come through the Center for Sustainable Innovation, which is located at the business school in Salford. Is where the big hub sits. And through the center or through the CSI, we get in touch with most of the projects in terms of biotechnology, especially sustainability. For all these projects, depending on the core of the project, we have an amazing team around us that will provide their expertise to address each challenge for different projects. But at the moment, our most proud achievement as a microjoule brew lab is we are sitting as a hub for different microbreweries now in the Greater Manchester area, which we are engaging and forming, starting collaborations to help them to overcome these challenges as an SMEs. So we have at the moment 7 brothers, Marble, Larhill as our commercial direct partner. We have Shearshaw, Balance Brewing, Cloudwater, Joseph Hall, Hip Hop, which is not beer for the ones that are into the healthy sector. Hip Hop is a company that produces kombucha, or this, and this is one of the things that I would like to highlight today. All this collaboration, building relationship have happened through the IBIC. This main engagement has facilitated the interaction between us and the microbrewery industry. We have submitted some proposals through the funding into the first stage with some of them, and not just them, and I have to highlight this because the company that we're using for the paper crumb are no breweries, but we have good relationship. We're trying to build a stronger and long-lasting relationship with Lubrizol as a chemical manufacturer company, which we are working together on that feasibility of using brewery waste to produce high valuable chemicals, more biochemicals that they could produce in a larger scale and then expand their portfolio of green products into the market. So this is one of our biggest achievements. This have happened for the last year, and the IBIC has helped us to expand and reach through these industrial collaborations. At the moment, we are part of the iCure program into their stage 1 with the aim of spinning out Microjoule Brew Lab from the university and keep offering these quality services across the food and beverage industry as much as the brewery sector. As I said before, this wouldn't be possible without the team that we have behind at the university, and especially our students. Our students are the ones that drive the lab, that are more engaged, that goes to the breweries, talk to the brewers, and come with initiative ideas that sometimes we don't have the time to come due to the multiple tasks that we perform every day. But it's rewarding to see their smiley faces, the way that they interact with the lab, and the way that they are engaged with the whole process. So without saying more, I'm going to leave you with Danny, which will explain more in depth what do we do with this brewery waste and what are the aims that we hope to achieve through it. So thank you very much. Stop sharing.
Okay, before we move on to Danny's talk, has anyone online got any questions for Ali about the work at Sulphurs, the Microjoule Brew Lab, or the work that she's doing with the breweries, or about her work with IBIC? Is there anything that anyone has any burning questions? So we've got Danny R. Hello.
Hi. Hi, thank you for taking my question. It's a bit of a strange one, but I work in a different area. I work in renal care, so like kidneys and disease and things like that. And I was quite fascinated by your conversation about the cleaning side of the tubes and the pipes. And you have to forgive me, I'm a complete ignoramus here. Okay, so I'm really bad. However, something that's been quite new to me is the issue that we are, for every single dialysis treatment for patients, we utilize 700 liters of water per patient per treatment. And going back to my ignorance, one of my considerations there is it would be a significant chunk of that would be the cleaning of the system and that kind of lack of ability to recirculate, to reuse that fluid, to keep that patient the fluid sterile, but also to maintain that patient safety. Have we ever considered where we could transfer this technology into the healthcare environment to maintain or to reduce water usage, especially in cleaning and things like that? Has that been a consideration as part of this, or is this?
The aim of this project was reducing the carbon footprint of their products to a more sustainable alternative. However, the approach was very interesting because was through the use of nanoparticle as a technology. So I don't have an straight answer as in that could be translated into those fluids, reducing the amount of water, which obviously will have a benefit for everybody. But that is an interesting point of view that I am sure my colleague Shalka, which I believe is in the room, will be able to expand in the further applications that nanoparticles or nanotechnologies could be used with that particular issue. So Shalk, if you're listening to me, you can put your email on the chat box. I'm sure Danny will be more than happy to expand this conversation. But yes, it's when a project starts into the application of one sector, likelihood is might be expanded to another sectors.
That's me saying thank you.
Thanks,
It's
Danny.
quite interesting. I think it's important to understand this about the sustainability. It's absolutely incredible. I was in an intensive care unit the other day, and they're all talking about it. I'm dealing with people in Zambia, and it's very much part of that conversation. But thank you for your answer.
Danny, Shalka just popped her email in the chat box, so feel free to reach to her and expand the discussion.
Hey, thank you. Any further questions? Otherwise, we will bring on Danny to talk about his work. So I'd like to introduce Danny Wales. He is a PhD student at the University of Salford, working alongside Ali and Natalie Ferry and other colleagues there. Danny, if you'd like to share screen.
Am I sharing it now?
Not yet, no. Hang
Oh,
on.
okay, right. I will put my, let me share the screen. Share. Center mode. Are you seeing that now?
Yes.
Oh, okay.
Perfect.
Great. Okay. Is that all coming through okay?
Yeah.
Right, great. Good afternoon. Welcome to my talk on making brewing waste valuable. I'd like to thank IBIC and my supervisors, Natalie and Ali, for the opportunity to speak today. Something not a lot of first-year PhD students ever get the chance to do. I'd like to preface all of this with the understanding this is very early in the research process, so please go gentle with the questions. I've still got a lot of learning to do. But whatever I can answer, I will answer. If not, I'll get back to you when I do figure it out. Just give me a little time. So let's move on a slide, if I can actually get this working. Right, so for anyone involved in brewing, these two products will be very familiar. However, for those that aren't, here we have hops and barley. They're a common ingredient of the brewing process, differing in quantities for beer, whiskey, gin making. After the brewing process, however, they are wasted, either disposed of in landfill at cost to the brewer or given away to farmers for animal feed. What if there's a third option? So when I was designing this study, I wanted to know basically how much raw material was available. Using data from Larkhill, Salford University's own brewery, and some statistical data supplied by Statistica, are used on beer, whiskey, and gin production, I was able to work out a rough figure of 94 million metric tons of brewing spent grains wasted per annum. By the way, if anybody wants it, the maths is all here in the PowerPoint. I'm more than happy to share it. I've spent a lot of time working it all out, so I've got it right this time. There's an awfully large figure for anyone to imagine. So to break it down into its simplest form, I'm going to leave the metric system for a moment, and that works out as roughly a fully grown African bush bull elephant every 2 seconds worth of brewing spent grains in the UK alone. And this doesn't even account for unlicensed home brewing. It's a lot. So brewing spent grains, essentially, like any plant material, it's a lignocellulosic material comprising of approximately 17% cellulose, 28% lignin, 20% protein, and 28% non-cellulosic polysaccharides. I'll be extracting my chemicals from the fibrous cell wall structure. So here we can see cellulose, hemicellulose. It can be broken down into a number of valuable industrial chemicals that span most industries. I don't have all the time in the world to go into all of them. So I'm going to concentrate on, if you look to the right, C6 column, 5-hydroxy methyl fluorophall. So cellulose structure, it's a polymer ribbon-shaped chain of glucose molecules, where those glucose units are connected by glycosidic bonds at the C1 and C4 positions. When this polymer is broken down, you can get a number of industrial chemicals besides sugars: lactic acid, gluconic acid, sorbitol, fluorophall as discussed, levulinic acid, ethylene glycol. I'm looking at fluorophall. So fluorophall, the value. We're looking at around 751 million US dollars by 2030. That's rising from 613 million US dollars in 2023. It's a lot of money we're playing with here. But where does that translate to? All of these industries rely on hydrocarbons from oil. And as we've experienced in the last few years, a change in oil prices can massively impact every part of our lives. What if we could replace that oil and use a recycled, infinitely renewable resource from what is essentially a waste product? There are so many everyday products that we can help change the price of, help influence a better, greener future. So I'm going to be brutally honest, right? I'm going to hold my hands up on this one. The technology already exists to break down lignin and hemicellulose, part of the plant structure, but, and there's the big but, it's costly. At present, we use high temperatures, well over 600 degrees C in most cases, high pressures, superheated fluids, strong acids, alkalis. All of these incur an energy cost or cause problems in the downstream process, which essentially translates to a financial cost. We need to find a better way. My aim is to use a process that uses less energy, chemicals, which in turn creates less pollution, and hopefully will be cheaper. Might even put some extra pennies back into the brewers' pockets. Fingers crossed. So, hemicellulose. It's a simple molecule, essentially, compared to lignin, which can be broken down into industrial chemicals. We can see the top 10 industrial chemicals here we can obtain from those sugars, feeding a renewable resource into a currently oil-dependent industry. I'm not going to spend too much time talking on this diagram, but again, if you want a copy, I'm more than happy to send it. I know that this is a bit of a complicated diagram, so please don't get overwhelmed, all right? Essentially, what I want you to take from this is that sugar structure can be modified using processes such as esterification, etherification, amination, oxidization, etc. If you look up in the top right, in the B section, you can see how changing these processes actually will, this is one for the chemists, that is, it changes how soluble that molecule is using a process of benzylation or carboxymethylation, which then affects the downstream processing, making it a very valuable change in a relatively simple format. So, our produced chemicals, we need to purify them, right? Okay. So the process is going to start with fractionation, purification to gain those cellulose, hemicellulose, lignin, etc., that we can process from that. It needs to be purified. So we have to fractionate it, we have to purify it. It's not a simple drop-in process. Hopefully it might be at one point, but we've got some work to go for that. After the extraction of hemicellulose, the molecule can be modified, which gives it a lot of control. That polymer chain can be reformed into different architectural structures, which give different applications, such as emulsifiers, hydrogels, aerogels, amongst many, many others, nanoparticles, etc. So the how is why you're all here. Listen. I'm looking at understudied organisms. So thanks to the work of people like Natalie Ferry's team, who've looked into wood-feeding insects, bacteria, and fungi, we have some targets. University of Salford has a swath of data that can be data mined, as well as NCBI, etc. They have that data there. Bioinformatically, I can find that data. I can find those enzymes. One target I'm looking at the moment is the fungi on the left-hand side. That's turkey tail fungus, brightly coloured. It's across almost every continent on the planet, and is described by many researchers as one of the best lignin and hemicellulose degrading enzyme producers there is, making it quite an interesting target. But that isn't the only one. There's 262 genes directly associated with lignin degradation in Klebsiella varicola alone. There are other enzymes found using data mining, which I've discussed. But essentially, I'm looking at lignin peroxidases, manganese peroxidases, lacases, and various other oxidases. All of these have effect on cellulose, hemicellulose, etc. My work heavily leans on this recent Nobel Prize to this team who created AlphaFold. I'm indebted to them beyond anything that I can imagine. Their work with Google's DeepMind allows me to take sequences, place it into the AlphaFold server, and form these 3D structures. I'm quite proud of the image on the right hand side. That is a image for a lacase from Tramedes versicolor that I've recently done. Here you can see the blue sections, which give a greater than 90% confidence in their structure, and the orange sections are less than 50%. The key behind this and being able to do this virtually is the costs. This creates a speedy assessment, limits the cost to the university. I'm sure that my supervisors in the university will be thankful for me not costing them a lot of money. But the data itself can be taken from BLAST, ProSite, etc. Once I have that data, though, and I've actually obtained the key enzymes which I want to work with, I need to work in vitro, because we can work in a virtual environment all we like. We need to translate that to the real world. So we need to express the enzyme of interest in a host organism or order it in purified. Depending on the costs and the viability of that will differ, but essentially buy it in. We're going to test the enzyme on brewing spent grains, do an analysis on that using fluorometric and colorimetric assays, and find out which one works the best. Once that's discovered, you need to start having a look at the different variables that can be influenced on this. So different concentrations, different pH, different temperatures, and using metal catalysts. Now, using some of the work by Rosa, one of the team at University of Salford, she specializes in metal organic frameworks, which are a great key for that biocatalysis process. And I intend to utilize that for building the overall industrial process and speeding things up. So how does this affect the chemicals and brewing industry? Well, waste that would ordinarily go to landfill can be valorized. We're going to reduce the carbon footprint, hopefully, and use one of the most abundant resources on the planet, which will also hopefully reduce the reliance on fossil fuels. Might even make fuel cheaper, but I can't make any promises on that one. Finally, I'd really like to thank David Newman at the University of Manchester for renewing my faith in the scientific process and peer review by making me relook at my maths, something I've mentioned before. But I have great faith in the peer review process, and it's something that I'm very, very happy for. So has anybody got any questions?
Thanks, Danny. That was really interesting, and I think the work you're doing there can certainly have a huge impact. I see there's a question from earlier from, is it Kushi?
Let me see if I can see it. Is it in the chat?
In the chat. Kushi's asking, are there any opportunities to get involved? So Kushi, is that with the University of Salford, with the microjoule lab, with the work that Ali's doing or Danny's doing, or with IBIC in general?
Yeah, if we know a bit more about Kushi, will be helpful. We are, at the moment, we are a lab within the University of Salford, which means most of our students are part of the Masters in Biotechnology program, where their final year project is done with the brewery industry. So I team up with my colleague Ian Goodhair. So across the two of those, get about 10 to 12 master students. And we found these partnerships with the brewery industry, and we send the students to work in close with them. We next intake for these projects is January. And in January, we will be sending students to Seven Brothers and Cloudwater as a commercial partners to develop the projects that they are interested with us. And in terms of internships, I am sure that most of these breweries will be glad to offer internships to help them with the brewery industry as a whole. If they are paid or not, that's a conversation to have with them. But I work close with them, so I know they are always in need for help and to expand and upskill the students into their facilities.
Okay, thanks, Ali. We've had a couple more questions in the chat for, I'm not sure whether it's, it looks like for Danny. Could this be applied to different types of plant waste? And do we have timescales on your work, for example, transitioning from in silico to in vitro?
So to James's question, I was actually typing a message to respond to that one just then. Yes, essentially that could be applied to any plant-based material in the fact that it is keyed to looking at those three base materials of the fibrous structure of plant material, therefore translatable across all plant material, hopefully. I'd say hopefully.
It could be things beyond sort of brewery grains.
Yes,
it could
yes,
be sort of agricultural crop waste or others. Yeah. Okay. And a question from Edward Flynn. Danny Wells, I've been growing some lion's mane mycelium on BSG from marble beers. Happy to have a chat if some samples of that might be useful to you. So good connection there, Danny.
Yes, definitely interested in that. Lion's mane I've been looking into along with the varicola due to some of the enzymes. I would be very, very happy to be in contact with that. It'll save Ali some money as well.
Yeah. Edward, if you're still on the chat and want to sort of put your contact details in there for Danny Wales to follow up with, that would be great.
Please,
yeah,
definitely.
Happy to do that, Danny. Yeah, no worries.
Thank you so much.
Okay, we've got, well, we're at 12:50 now. We've probably got time just for one more question, but if you have any sort of burning questions post-webinar that you want to follow up with, you can contact us at ibic@manchester.ac.uk, or we're online, we're on LinkedIn, or you can contact directly the Microjoule Brew Lab to speak to Ali or Danny.
I'll get into typing those responses out as well for the ones I've
couple more
spotted.
messages. Oh gosh, right, okay. So another from Danny R. Tell me how you see this research impacting food science in 10 years.
10 years time, I would hope that it's a way for us to rethink how we're using our green waste. It doesn't have to just be something that's used for compost. Potentially it could be used as a valuable resource that could be collected. You know, imagine your bin man becoming an extra in stream for the chemicals industry. It's an entire possibility. One that might need some tweaking, but it's possible.
Okay, and then we have a question from Sabrina Zaney. Daniel, how do you deal with the variations in the waste stream and the byproducts affecting production of your target products?
I have to be honest, that's going to be an interesting one to cross. At present, the variations are quite limited due to the pretty stable quantities that are involved with brewing. It's always going to be a vaguely similar construction of distribution, sorry, of products, but once moved into differing plant products, yeah, I can see there'd be some hurdles. It would be very interesting to see. I'm sure it's one that can be overcome, though. I guess we'll cross that bridge when we get to it.
Okay, thank you, Danny. I think that's all the questions in the chat. I think Ali has responded to the final one there on the MSc. So I would just like to say on behalf of IBIC a big massive thanks to both Ali and Danny for their time today to present. I hope you've enjoyed our first webinar, and I'd like to just give a quick shout out to the next one we've got coming up. Jen, if you're on, is it on the 21st of November?
It's on the 19th of November.
The 19th. Oh yeah,
it'll be at midday on the 19th of November.
Yeah, so we've got Dr. Neil Dixon coming to talk to us. He's from the Manchester Institute of Biotechnology. So if you would like to sign up, the sign up form I think will be going out very shortly, if not available already. And you can look on our events page as well, or contact the team just to find out more. Okay, thank you very much, everyone, and wishing you a lovely afternoon.
Thank you.
Thank you, everyone.
Thank you. Bye-bye.
Thanks, bye.